Welcome to Oncology Morning Commute, a review of the science behind multi-cancer early detection tests. Morning Commute is developed by Projects and Knowledge powered by Kaplan and is part of a continuing medical education series. This independent CME-CE activity is supported by an educational grant from GRAIL. In the second episode of our podcast series on early cancer detection, Dr. Christopher Mason and Dr. Pashtun Kassi take a closer look at multi-cancer early detection tests and the groundbreaking PATHFINDER study that so far is showing impressive data. Information about the faculty and disclosures can be found at morningcommutepodcast.com/cancer early detect 2. You can use this link to receive your credit and evaluate this program. The URL can also be accessed in the episode notes. Dr. Mason is a professor of genomics, physiology, and biophysics in the Department of Physiology and Biophysics at Weill Cornell Medicine in New York City. Dr. Kassi is Director of Colon Cancer Research at Weill Cornell Medicine and Director of Liquid Biopsy Research at the Englander Institute of Precision Medicine, also at Weill Cornell Medicine in New York City. I am your host, Candace Hoffman. Dr. Mason will begin our discussion.
All right. Well, welcome back, Dr. Kassi, to our discussion on multi-cancer early detection testing.
Well, thank you very much for having me.
So we are going to jump back into this fascinating field and the development of the technologies that enable us to pick up not just one or two cancers, but sometimes several dozen cancers with one test. And, I want to jump into a little bit about how they work and, what's happening once you get the blood drawn. So, as I think most people in the audience know, of course, you can go get blood drawn for all sorts of work on what cells are in your blood or biochemical profiles, as a general measure of health. And so the vision that's come up for these multi-cancer early detection tests is that anytime you go get a blood drawn for a normal physical, you could potentially, especially if you're high risk, throw this in, and has to be kind of the standard of care where you just want to keep an eye out for anything that might be an early stage cancer. And the way that works is because, floating in that tube of blood, think about when you go get your blood drawn and you squeeze your fist and you look down at your arm, or maybe some of you look away, it depends on who you are, but you're getting the blood drawn. And into that tube are, of course, cells and of your blood, your platelets. You can see antibodies that are there, white blood cells, dendritic cells, all these different kinds of immune and functional cells in your body. But also floating in the bloodstream, in the plasma, you'll also see fragments of DNA that are sometimes still packaged up in what's called chromatin, where there's DNA mixed with proteins that are still packaged together, and they're stable just long enough so they could be picked up, and you basically take that tube, and you can see what's essentially inside your cells versus what's floating in your plasma, or also in other small pockets, what are called exosomes. But basically inside your blood is this entire really snapshot of every tissue in your body, because all of your cells are dying and then being reborn throughout all of the different organs and tissues of your body. And this is a continual diurnal process that you're shedding cells, shedding DNA, shedding RNA and proteins. And in that tube of blood, though, is this really whole body molecular scan that anything that was recently exuded or decayed or died and went into your bloodstream, you pick up this snapshot. And what you do is grab those fragments of DNA. We'll talk about other methods as well, but a lot of the tests just look at the DNA, grab them and purify the DNA out and sequence it, and then can take those fragments and map it back to what is called the human genome reference, which is where do you normally see open or closed DNA in different cells in your body, what this is called epigenetic signature of the state of those cells. And when you map it back to the different areas of your body, you can tell which cells were contributing that signature. Was it from pancreas? Was it from your bone marrow? Was it from, it could be an ovary, it could be any part of your body that's contributing little bits of DNA. And you build this whole body molecular scan. And you also then look for mutations. Is it when you look at the A, C, G, and T of the actual genetic code, if it looks the same as your reference genome or doesn't look like it has any mutation, then that's fine. But if you see mutations and you see enough of them, you can start to infer that you have mutations in a gene that might give you a risk for cancer, or maybe it represents the cancer. And you can tell to a large degree where it came from in your body based on where it lands in the genome, and also the fragment size tells you information about potentially how aggressive a cancer might be, what's called fragmentomics. And so these are some of the basic chemistry and functional work that goes into the test. But, so Dr. Kashi, with that as a backdrop, what's exciting you the most in terms of, once we get these profiles, we do the sequencing, we do the mapping and the alignment. What have you been the most excited to see in clinic, and what do you see so far for, say, insurance companies looking at getting excited to maybe do coverage? Are they waiting back a little bit to see how it goes? What have you seen in the clinic so far?
I think as an oncologist, unfortunately, we often see the late stage cancers where it's already having an impact, and often sometimes or it's not a curable scenario anymore. So the promise of these tests going into early detection or screening, I think that's what's really fascinating and brings up a huge promise, because it can do what they call like the stage shift, meaning that as opposed to catching a cancer in latter stages when it's already not curable in some scenarios, if it's stage 3 or 4, versus if you caught the same cancer at a very early stage, the outcomes or the odds of achieving cure, or as they say, 5-year survival in terms of being alive, they are very different. So I'm more excited about the fact that these assays that initially were crudely looking at mutations and DNA are now looking at these other aspects of the cancer, like you pointed out, in terms of methylation and other signals that not only point out to the presence or absence of cancer, but also with these different patterns and signatures, you can also identify where these cancers are coming from to a fair degree of accuracy.
Yeah, and I think that is what's extraordinary is, each one of these fragments, of course, has the genetic code, but has what's called methylcytosine, which is when you have a methyl group attached to it. But other studies have also looked at hydroxymethylcytosine, which is just adding another hydroxyl group to that methylation signature. And what I think is extraordinary is we've begun to tease out, as you were just saying, that there's all this additional information, not just about the mutations or the fragment size and where it maps, but then these small chemical tweaks onto each molecule, it themselves can tell you whether it came from really an open or closed part of the genome, or something that had a signature of rapid cell division, indicating it might be a more aggressive cancer. And so you get these really interesting signatures that have really become more prominent, I think, in a lot of the tests on the market, and a lot more research into this space as well. And so,
to your point regarding the aggressiveness, I think one aspect that I often like to highlight, besides the test, I think it's also important to remember what's happening before whatever tumor-derived content is going into the bloodstream, going into the tube of blood. The shedding, the aggressiveness of the cancer, the type of cancer, that could be different across different tumor types. And some of these studies are also showing that the same assays were having a different performance. So it's not just how good the tests are. We have to kind of put that into context with what cancers are being detected, what cancers are being missed, what cancers are creating a similar signal, as noted in some of the studies with one of the multi-cancer early detection assays. Cancers that were, for example, driven by a similar mechanism were also giving, not surprisingly, similar signals. And for those who are following the development of these assays, I would like to point out that these assays are continuously being refined, and the data that you might be looking at from a few years ago may or may not be relevant to how that assay may perform right now in clinic.
Yeah, I think because there's a continual improvement of the assays, which is great, but then it makes it hard to compare a bit more on the science as well. So a lot of people think of these fragments of DNA, but often they are either captured, like a Velcro, where you want to grab a piece of DNA before you sequence it. So there's an enrichment process for some of the tests on the market. Other ones do an amplification, where you actually target it with primers, or basically a way to also, in a sense, add little mini Velcro to your molecule and amplify it to make many copies. But those are two different protocols that have pros and cons themselves. And so what you'll pick up, what today might be a panel that has 50 genes in it, but if you come back to the same company 2 years later, they might have a panel that has 280 genes. And so you have a lot more. But it's basically, it has different probes, different molecules that are doing this Velcro and capture, and there are different amplifications. So the tests do change. Usually they get better, but sometimes you may no longer have a gene that's present in the panel if it turns out it was too difficult. So that this is a continual improvement, a constant gardening that has to be done to make sure the tests get better. But that does change how well you can compare them across time. The other thing that's, I think, really interesting in terms of the science and the biology of what's happening is when you take the liquid biopsy, when you take this test, what you'll see will depend on what happened recently to that patient. So if they had recently and been doing a lot of exercise, you'll see a lot more vasculature that has been in motion versus someone's been much more sessile. They wouldn't have much blood literally moving, which could change what you'll pick up. Or if someone just had surgery, that will lyse a lot of the cells that were right near the point of the surgery. So suddenly you can see a lot more of the tumor, but then it can go away quickly. And then so you'll get a discordance between what's in the blood versus what's maybe in the tumor later on, but you'll get a lot more concordance and see it right near the point of surgery. But you basically, this question of which cells are being broken open and lysed and dying at different points and what you're doing with your life, or did you just have a wrestling match basically, or did you get into a bar fight? I actually have no clinical correlates to bar fights and cancer detection. That's just off the top of my head. But it is a factor that may have some role in terms of how many cells have just recently lysed in your body. So
very important aspect. And like you mentioned, there's a lot of, before even the analysis goes into the test, what we call like the pre-analytical variables, meaning things that can affect the performance of the test. You brought up the surgery aspect. That's very relevant, when we are looking for leftover cancer or what we call minimal residual or molecular residual disease in the immediate postoperative period. There's a lot of background noise from all the healing that your body is undergoing. So you're looking for the needle in the haystack, and it's a bigger haystack in the immediate postoperative period. And as these assays are being used for screening and detection, just like in sometimes with these imaging modalities like PET-CT scans and stuff, if somebody just had an infection or is undergoing through some other medical problem, we often say that's probably not a good time to get a PET-CT scan because you may get a false signal. I think we would also learn over time as these tests and these trials and ongoing studies mature as to what real world considerations need to be factored into as you're getting the blood drawn. As you mentioned, things simple as an exercise or somebody who had a smoke or even the time of the day. There's a lot of interesting data and literature about variations into this that can happen. But despite that, the fact that you can detect these things non-invasively through simple blood draws, I think it's very fascinating and promising.
Yeah, and this does link to the ease of which you can do it, getting a blood draw I mentioned earlier from anywhere. But this does also link to, we'll jump, I think, back into some of the study statistics that have come out recently. But just to jump on the point of the ease is, if you think of patients in rural settings or lower economic status, people with lower resources, less resources available to them, they, it is nice if you can just stop by a place really close to your house, get a blood draw or put it in the mail. Or even some of the testing I've done was a tube came in the mail, and then I went to a phlebotomist and put it back in the mail. But I even just recently saw a non-invasive prenatal testing test came out this week that you can take it home, right? So I think there is a nice burst of convenience happening that I think will help for accessibility to these tests. Of course, they're still a bit expensive for most people, and so I think that hopefully will come down. But we've seen on the technical side, the cost of sequencing is coming down. The cost of these assays are now getting much more standardized, and so the tubes are now being made by the tens of millions, so they're getting cheaper at scale. And so I think we'll start to see more of deployment and ease of access in the coming years.
And with the different healthcare models and systems, there are parts of the world where it is not the insurance or the healthcare or the whatever health system that pays for the test, and it might be something that the patient may be factoring into out of pocket as we go along and develop these technologies. Like you mentioned, as these things get cheaper, I do think it will fill a void, and for the existing tests, as some of the data and the papers also cite correctly, that for the ones where existing assays are available, it's not a question of either or. I think it will complement each other.
Yeah, absolutely.
But for the fair majority where there's nothing, I think it opens up an avenue for screening and early detection for a lot of these aggressive cancers that don't have a cure in latter phases of diagnosis.
Yeah, yeah, there are very small sequencers that can fit in even in your pocket. So someday, maybe in the future, you could draw your own blood, sequence it in your own kitchen countertop, and then get a report. But that's probably a bit more futuristic. But of course, I think that'd be wonderful. But in any case, but that would definitely not be a CLIA test. It would be it's a homebrew test. So you'd worry about accuracy and thoroughness there, of course. But but I want to jump, in some time we've got left in this podcast a little bit, is to some of the studies that have come out that have really started to examine at a large scale what we think we can learn when we start to have tens of thousands of patients that have been examined, and we're seeing large international efforts to track what we can pick up by looking at people longitudinally over multiple years in some cases. And so one of them in particular is a trial that was just recently released. It's called the Prospective PATHFINDER study, which did use a blood-based multi-cancer early detection test and looked at early cancer screening for over 6,600 individuals that were at least 50 years old and were at a higher risk for cancer. So they used some of the methods we just described here in terms of sequencing and mapping those fragments of DNA to look for the cancer signal origin, or trying to figure out basically where in the body the cancer is based on this test. And basically what they found was they wanted to determine first question is, how many people actually had something that looked like it was a positive hit? And so basically out of those 6,600 people, there were 92 participants who actually tested positive. And so this gives you some, you'll see about we've heard in the literature, about 1%, 1.3% of people you'll get something that there's a hit, and say, okay, well, of those patients, how many of those 92 were actually diagnosed with cancer? And of those, it was about a third, 35, were diagnosed with cancer through the follow-up. And I think that really the most exciting about the study is that of those 35, narrowing it down to just those 35 people who had confirmed cancer, 71% of them were of the cancer types that really don't have routine cancer screening. You think of like mammograms or PSA for prostate cancer. For a lot of the cancers that were picked up, the majority of them, there are not routine screening options for those cancers. So I think this highlights some of the value. And I think one of the really exciting things also is that for those confirmed cases, 48% of them were stage 1 or stage 2 cancer. So meaning we could pick it up earlier. And so that's, I think, some of the exciting overviews, but there's also questions about, what's the false positive rates and what do we see for the positive predictive value? And what we saw in the study was a positive predictive value of about 40% and about a 1% false positive rate. So I'm curious, Dr. Kashti, what do you think about in terms of these rates and what you see for other tests? And I think they've been getting better in the field, but what have you seen so far in a clinic?
Well, I think you bring up an important point regarding the results, and especially, it's somewhat concerning and sobering to note that, we only have screening for 4 or 5 different cancers, and also those screening modalities are not necessarily perfect or convenient. Example, colonoscopy for colon cancer or mammograms for breast cancer. So the blood-based testing detecting cancer for obviously complementary for the ones that is existing screening, but also as you mentioned, in the patients where they had detected, a lot of these were for cancers where there's no screening at all. I do think it's of value to note, as we were discussing earlier too, that there is continuous learning and refinement that's happening with these assays. In some of the initial work published regarding the study from the same group, they've noted how refinements for site of origin were happening. In some cases, they haven't had a diagnosis yet, and is that still because we haven't found the cancer yet, or is that truly a false positive rate? In the big scheme of things, like you mentioned, I think these numbers are of value to note that, if you are to implement such sort of a testing platform, you are looking at, no more than 1 to 2% of patients who would have a positive signal that would need downstream further testing and evaluation, and some of the subsequent studies that will integrate downstream testing. I think those are also questions and things to ponder from a payer perspective, that obviously it's not just the test that you're paying for, but then you are to do follow-up workup and evaluation or further testing in the ones who are positive. So what would the math look like there? I think those are bigger questions that the community and CI and the scientific community is trying to answer.
Yeah, because you really want to gauge the utility, what's the bang for your buck, or the efficiency of the test. And we can't spend every dollar for every test on every patient, but really figure who's the high risk, or medium or low risk, and even tailor, I think, some of these screening tests and these early cancer detection tests towards the best test for and the best frequency for the right patient. Great. So just in the last couple of minutes, there's one other study I wanted to mention, which is from the Cheng-Kung Medical Foundation, where they also were looking at how many malignancies get detected by having these cancer screening tests. And I think what's fascinating there is we see different cancers have a higher or lower sensitivity in even being able to be picked up. And for example, prostate cancer compared to pancreatic cancer, those both have pretty high sensitivity. Whereas if you look at head and neck cancer or kidney cancers, look on the lower end from their study, or even skin cancer. But some of them make obvious sense though. Skin cancer and melanoma might be in the outside of the epidermis and not be as picked up as readily because there's not as much vasculature pulling what might be fragments of that cancer into the blood that you could be picked up. But I'm curious here in the last minute or so, any thoughts on the heterogeneity of detection between different cancer types, and maybe any ways we could get around that, say, frequency of testing or other modalities?
Yeah, the study you mentioned, I think that's not entirely surprising or new to us. In oncology, we use these tumor markers, which are often what we call glycoproteins, like the ones that they used in the study with alpha-fetoprotein, PSA, CEA. So we know that certain tumors, and often tumors that are from glandular origin or that make glands, like pancreas or colon or breast, that they often make these markers, but they're not entirely specific nor sensitive. So even today in our clinic, probably half the people had some sort of tumor markers checked. So we don't check it for every single type of cancer, and they are not necessarily elevated in some cancers like skin cancer and stuff. So that's not surprising. We don't see the performance of that in, let's say, the head and neck cancer or some of the ovarian cancer group. But like, for example, the same CEA is elevated in breast cancer, stomach cancer, colon cancer, and it's not entirely unique to one cancer. So I think their strength, I guess, was looking at all these different common glycoproteins together. But building upon this, what I can tell you is, following this field, there are larger efforts, not just looking at methylated DNA and other aspects that we mentioned, but looking at not just these 7 or 8 or 10 glycoproteins, but looking at hundreds of these in a very small amount of sample using bigger platforms, which are already kind of in study, and we've presented some of the data as well with some of these collaborations. So I think it's a little crude and very different approach to the methylated platform, and is of value to see how it could further complement the existing platforms.
Yeah, that's a great summary point. I think we know that it could be proteins, could be metabolites, could be mutations. All these different modalities of biology are there and can tell us something about the cancer, if it's there, or its progression, or what's happening in the body. Every molecule tells a story. We just have to listen to it and then hopefully use it. So I think that was a great closing thoughts. And thank you very much, Dr. Kasti, for joining us today on our oncology morning commute. And to wherever your commute takes you in the audience, I hope you don't have any mutations more than you did 5 minutes ago, or that if you do, that we can pick them up quite readily.
Remember, to receive your credit and evaluate this program, please visit morningcommutepodcast.com/Cancer Early Detect 2. You can find all of the episodes in this series and all of our other podcasts on your favorite podcast streaming services, or download our Morning Commute app. Thank you for joining us today.